This is our research plan in which we are aiming to elucidate the neural underpinnings of place learning through task-dependent functional connectivity and transcranial magnetic stimulation. Successful navigation relies on both egocentric processing, for example, the church is on my left, and allocentric, for example, the church is to the north of the park. Previous research has indicated a complex network of brain areas involved in these spatial reference frames. However, the precise neural mechanisms and causal roles of these brain areas are not well understood. Here we will use fMRI and TMS to further our understanding of these neural mechanisms. To do this, we will use a place learning task as demonstrated in Figure 1, in which the participants are placed into an arena and must first walk and pick up a flag in the training stage. They are then returned to their starting location and must return the flag to where they believed they picked it up in the testing stage. After each trial, they then receive feedback indicating their accuracy on the task. Crucially, in this experimental design, we can manipulate the participants' reliance on egocentric and allocentric cues by manipulating the experimental arena, as can be seen in Figure 2. To identify the brain areas involved in these processes, we will have participants complete a version of this place learning task inside an MRI scanner while we conduct resting state functional MRI scans. To analyse this data, we will look at using a task-dependent functional connectivity analysis, in which we conduct a general linear model with task condition as an exploratory variable, and we will use brain areas such as the hippocampus and precuneus as seed regions based on previous research. We expect to find differences in functional connectivity between the allocentric and egocentric conditions and relative to the control condition, indicating the brain areas and networks involved in these spatial reference frames. Once we have identified the implicated brain areas involved in these reference frames, we will then look at investigating their causal roles using TMS. In this experiment, participants will take part in 2 to 3 sessions separated by at least a week. Within each of these sessions, we will deliver either excitatory or inhibitory theta burst stimulation to the brain areas identified from the results of Experiment 1. Following this, they will then complete a desktop version of the place learning task. A within subjects ANOVA will be conducted to look at the effects of task condition and stimulation target on location error and path data. We expect to find that theta burst stimulation will differentially affect performance on the place learning task depending on both the stimulation target and parameters. A combination of these 2 experiments will allow us to not only identify the brain areas involved in egocentric and allocentric processing, but will also crucially give us knowledge of their causal role.